My lab has a long history of studying how cells respond to genotoxic stress. And the whole basic goal of our approach was if we could understand how cells are responding to genotoxic stress and the signaling pathways and DNA repair pathways that are activated by such stresses, we would be able to better influence how current chemotherapy drugs work, particularly chemotherapy drugs that cause genotoxic stress. And all the classic ones that you know of kill tumor cells by inducing DNA damage and or blocking DNA replication. And so we went about, back when I started my lab, saying let's figure out some of these pathways, and we particularly focused on checkpoint signals, which, as you will see, are signaling pathways that can activate apoptotic pathways, but in tumor cells they don't do this so much. Instead, they activate cell cycle arrest, and it turns out that for the most part these pathways help tumors survive various types of toxic stresses. In addition, checkpoint signals also regulate DNA repair. So although they were thought to primarily affect the cell cycle at the time, it's since been discovered that they affect all kinds of cellular responses. Two chemotherapy, primarily one chemotherapy drug, and my slides are being cut off, I see, but this says 5-FU, 5-fluorouracil. It's one of the oldest and also remains one of the most widely used chemotherapy drugs, primarily. And it's active in many tumors, including widely used in colon cancer, breast cancer, used in pancreatic cancer, and other upper aerodigestive tumors. The other drug I'm going to tell you about as we go along here is one called fluoropyrimidine, rarely used today, but it's actually an FDA-approved treatment. And so the first thing we did was ask what happens to these pathways when we treat cells with 5-FU and fluoropyrimidine, which we sort of did as a control, and said what happens. And so we threw 5-FU and fluoropyrimidine on the cells for various periods of time, these are hours, and as well as hydroxyurea positive control for this pathway. And what we saw were dramatic activations of CHEK1 and CHEK2 by both drugs in both cell lines, and although the kinetics differed and the relative rates of the activation differed, they're pretty much, this is what we see when we do these types of experiments. And so we could also show that both drugs were inducing DNA damage, and we did this by looking for a marker of DNA damage, which is actually phosphorylated by ATM and ATR. And you can see that 5-FU is causing DNA damage in both cell lines, just like hydroxyurea does. And so from this, we concluded that fluoropyrimidine is clearly doing this DNA damage pathway, and so we should study fluoropyrimidine. 5-FU, we're not studying it, but I suspect it's killing cells. Even though it's causing DNA damage, the primary means it's killing cells is through RNA damage, and like I said, we're not going that way. We're going this way, and we're asking what happens with fluoropyrimidine. So our conclusion from this is that this is a really potent combination. Okay, so with that, I can summarize that's what we've done. I can summarize where we're now going. What we want to do next is we want to actually look to see and evaluate the roles of various DNA repair pathways and checkpoint pathways to this drug combination, because we actually don't know what kind of DNA damage occurs when you put PARP inhibitor together with fluoropyrimidine. And so it could be that there's certain repair pathways under checkpoint pathways that become very important with that drug combination that might help us understand how the cells are responding to that and dying. The other thing, and primarily the funding that we're using from the Hirschberg Foundation, is to assess toxicity and anti-cancer activity of fluoropyrimidine plus a PARP inhibitor compared to fluoropyrimidine alone in a mouse xenograft model of pancreatic cancer. We're also hoping in the future to look at this in the more elegant models of spontaneous pancreatic cancer in mice. And finally, we're actually working with our clinical colleagues to develop clinical trials to test this in patients.